Inside this egg is a ticking time bomb. In just 4 hours, a single nucleus has to clone itself into 6,000 copies. Every piece must be ready perfectly before the egg seals those nuclei into separate cells. And if it fails, well, there is no backup plan. It doesn't just get a leg wrong. The whole thing collapses into one large chaotic mess. So, how do thousands of individual parts move as one when every second counts? Okay, but wait, why the rush? Because for a fruit fly embryo, the world is a nightmare. Your mother dropped you on a piece of rotting fruit. You're surrounded by predators. Every second you spend as a helpless egg is a second you're on the menu. To win here, the fly embryo makes a radical choice. It abandons the standard rules of biology. See, most embryos, like fish or human, take their time building membranes around new cells. But walls take time. So the fly makes a high-stakes decision. It ditches the walls altogether. No walls, no separation, just 13 rounds of nuclear division inside a single cytoplasmic ocean. But that brings us back to the paradox. There's no brain in charge, no leader. So how do thousands of nuclei stay perfectly synchronized all this time? Well, let's wind back. Look very closely. Notice something strange? Division doesn't happen all at once. It moves like a wave from the poles to the center. But why a wave? Why not all at once? You might think it's like falling dominoes. One nucleus signals the next. Could be, but there's one big problem. This embryo is gigantic, 100,000 times larger than a normal cell. So a chemical signal would be way too slow. By the time it reaches the middle, the ends are already dividing again. It would be total desynchronization, total failure. So what's really controlling this? Surprisingly, from the moment each nucleus is born, they all have their own super precise timer. The alarm in this case is a protein complex called CDK1 cyclin B. They're the green light for division. Okay, hold on. If they all have the same clock, why aren't they then all dividing perfectly together? Why does a wave even exist? Well, here's the twist. After each cycle, cyclin B gets destroyed. The clock resets. And because the first nuclei cluster in the center, they lower the local cyclin B supply. But at the edges, levels stay high. So in the next cycle, the edges hit the threshold first and then the middle follows. It's a mesmerizing wave, not caused in real time, but robustly pre-programmed cycles earlier. But wait, did you notice something's off? According to this model, the nuclei all stay in the middle. In reality, the nuclei do start in a clump, but they end up perfectly spread out along the membrane. How did that happen? Well, have a look at this earlier time point. Watch this. The embryo looks like it's glitching, no? It's pulsing. But why would a system obsessed with precision introduce chaos? Remember our go signal, CDK1 cyclin B? Well, there's also an off switch called protein phosphatase 1. And every time a nucleus finishes dividing, this protein ripples outward. When it hits the surface, it activates myosin, the same motor protein in your muscles. The result? The embryo literally squeezes itself. It's exactly like pressing the middle of a tube of toothpaste. The internal fluid, the cytoplasm, is forced towards both ends, dragging nuclei with it. And wave after wave, the nuclei get pushed to the edges until they are perfectly spaced. Once they are spread out lengthwise, the nuclei move out towards the surface. It's the emergence of a biological map. Alright, but here's where the physics of this embryo get really strange. It's actually pretty hard to spot, so I first want to show you this next video. Can you see what is hidden in plain sight? Notice that with thousands of nuclei together, none of them ever crashes into each other. Scientists actually took these nuclei out and put them in a dish to see. Turns out, these things are obsessed with personal space. No matter how much room they had, they always pushed each other to fill up the whole space, just like in the embryo. And the most bizarre part? They always ended up exactly 28 micrometers apart. It's like they have a hard-coded personal bubble. Scientists even tried to push them into tiny microchambers. And guess what happened? They didn't shrink or scale down, they just stayed too close together and turned into a tangled mess. The result was a total disaster. So, what creates this invisible force field? When a nucleus divides, it shoots out thin tubes called microtubules. They look like a starburst, and they're called an aster. You can think of them as mechanical bumpers. When two get close, they physically shove each other away. This repulsion forces the nuclei into a perfect hexagonal grid. But here's where it gets impossible. Scientists created embryos with no nuclei at all, just asters. No DNA, no control center. And the system still organized into a perfect grid. Now imagine something goes wrong. A nucleus disappears, a gap opens. Does the system collapse? No! The surrounding asters rotate towards the empty space. They rebalance and fill the gap instantly. It's a self-healing grid. After 13 rounds, time expires. Now, and only now, the embryo starts building membranes. Each nucleus gets sealed into its own cell. Because of this high-speed miracle, every one of those 6,000 cells knows its exact coordinates. They have moved from a single point to a flawless biological map, ready to become eyes, wings, everything. Now, you can find all the research papers below. But here's the real question. If a single egg can coordinate thousands of units without a leader, what kind of invisible systems are going on in your body right now? Well, just watch this video to see the strange hidden life of your mitochondria.